Last time, we were looking at a BioCyc protein page, in this case, an enzyme. This time, we'd like to look at a reaction. So from the THRA page, let's click through to one of the reactions it catalyzes. Here we are on a reaction page in a BioCyc database. So what's in the page? Well, up here at the top, we have the name of the organism, the name of the reaction. In the case of reactions that have EC numbers, we label them with the EC number. Down below, we have a cross-species comparison button. This is a way that you can compare the presence or absence of this reaction across multiple organisms, as we'll address in a future webinar. Below that, we have a breakdown of the classification of this reaction, again using the EC hierarchy. Then below that, we have a list of enzymes and the genes coding for those enzymes that catalyze this reaction. And down here, we have the reaction itself with structures. So we have L-aspartate turned into L-aspartyl-4-phosphate. We've abstracted out the structures for ATP and ADP on the assumption that one, you know it, and two, you can click through and see the structure. And there, if you needed it, is the structure of ADP. Continuing down from the reaction here, and you'll notice that as I mouse over stuff, it tells me what I'm mousing over, the compound, its synonyms, that lets me know it's clickable. Continuing down, we have the official EC name for this reaction, if there is one. We again have the gene reaction schematic, and we have unification links, in this case to Brenda and enzyme, which are both very useful resources. You notice there's not a whole lot on the reaction page. It's very clean, and this is the point that I was making in the last part of this series about the purpose of the enzymatic reaction frame. This is, if you will, the platonic ideal form of the reaction. This is not about how an enzyme carries out the reaction. The reaction page is about the mechanical aspects of the reaction. You take in L-aspartate, you somehow combine it with ATP, you get out L-aspartyl-4-phosphate and ADP and a proton. That's that. When you go over to an enzymatic reaction section on a protein page, then you learn the nitty-gritty about how that particular enzyme carries out that reaction. Does it have cofactors? What are its KMs like? Etc., etc. So that's the point of that distinction. So with that in mind, there's not a lot more business going on in a reaction page. And that brings us to our final installment in this series, where we take a look at compounds. So I hope you'll come back for the final part of the Intro to BioCyc webinar series and take a look at what's in a BioCyc compound page.